Hello. Now, this is a review of practical knowledge and understanding, and this is our practical skills. Now, we will talk first about the types of variables we encounter in the lab. There are three types of variables that we encounter: the independent, the dependent, and the control. In order to explain them very properly, we need to put a reaction in front of us and see how that is affected by the three variables. Now, this is a reaction that involves an H2SO3, which is sodium thiosulfide, and also hydrochloric acid. Now, this reaction produces a precipitation, which is sulfur in the form of solid. It also produces NaCl, H2O, and SO2 gas. So, we can either measure the sulfur or the SO2 gas, and we can see the reaction as it progresses. Okay, how does that have to do with our independent? What does that have to do with our independent, dependent, and controlled variables? Well, let's see. If you want to carry out this reaction, you might want to minimize all the errors. So you might want to do it more than once. In this case, you might want, for example, for the HCl volume to be constant. Regardless of the concentration of HCl, you want the volume to be constant. Another way which we can control variables is by controlling the room temperature. We carry out all of our reactions at room temperature because we want to control it. If we were carrying out a very sensitive reaction in our labs, we might need to control further things like dust or even the air flow. So there are several things we can control, and those variables that we can control are called controlled variables because we must keep them constant to ensure that a fair test is being carried out. Okay? So whatever you choose as a controlled variable, it must be kept constant. Okay, wonderful. What about the other things? We know that we change one thing and measure another. So what does that have to do with variables? We have two other types of variables. We have the independent variable and the dependent variable. The independent variable is the one being investigated, and we change it constantly, and we can choose different values of it. For example, we can bring different values of concentration of hydrochloric acid. Now that would be an independent variable because we chose it. We set it, okay? We chose different things, and that is the independent variable. Probably we are studying the effect of HCl concentration on the rate of forming solid sulfur. So what does that make our dependent variable? Our dependent variable is the one that we need to measure to judge the effect of changing the independent. So, for example, we might set different concentrations, and that would be our independent, and measure weight or mass of sulfur, and that would be our dependent variable. Another way which we are familiar with is changing, for example, the time is changing the time in order to find out the reaction rate. So if you place a reaction vessel on a hot plate, you might want to change the time in different intervals and see how does that change the color of your reactant. Now, usually the questions where you have independent and dependent are set like this. How does the independent variable affect the dependent variable? In order to understand any question properly, you need to know which one are you investigating. What are you doing exactly? So I suggest before you go into any practical exam or any practical lab or anything with practicals in it, is just to take a seat and read the question properly. Understand what is being investigated. Understand what is being changed. For Cambridge examination skills, which is paper 3, you need to follow the instructions and carry out an investigation. Now, the investigation is of unknown substance or a mixture of substances. So, Before carrying out any test, always read through the instructions. Now, because when you test unknown substances, it will require you to describe your observation details. And this amount of details that you need to know, what you need to observe is probably written. So if you need to observe, for example, volume of gas being released or temperature, you need to know what you are measuring. You need to know what type of investigation you are carrying out. Another thing, you need to know that whether or not to make up a table or a small investigation. For example, one way is the enthalpy, which we are familiar with, I hope. In the enthalpy test, we have two types of questions. We have one question that is asking us to measure temperature every 30 seconds, and we have another test that is asking us to measure temperature at the highest and lowest. That would mean that for one type of question, we need to make up a very long table and then draw our results. For another type of question, we need to make up the initial, so T initial, and T final only, and to get these 2 points. You might be asked which one is more accurate, or which one do you prefer to use. That would be up to you. You need to have a clearly stated reason for any explanation you have. Now, in your results, you might have different intervals. An anonymous result is a result that does not follow an established pattern. So if you need to draw something and you have this, for example, and then you have these types of points, and you have 1 point here. This point is anonymous because all of them would follow a certain pattern, and you can draw all of them as a best fit line. Okay, there are things that you need to remember before going into your paper 3 examination. Is when you describe a liquid or solution that is not colored, you need to say colorless. If it's transparent and not colored, you need to use the word colorless. For example, copper 2 sulfate is transparent, but it has a blue color. So you cannot say that it's colorless, but you might want to say that it's clear. Clear in the definition of Cambridge means transparent. Now, if you have a solution that is white and opaque in chemical test, it probably contains a fine suspension of white precipitate. If you are carrying out a titration, you should repeat the test until you have 2 titers within 0.1 centimeter cube of each other. Ideally, you should be able to do this. If you can't, be patient. If you have a titration, please remember that you need to do a rough value. Now, this rough value will help you establish where your actual endpoint lies. For your presentation, if you need to present numerical data, values, or observation, make them all in a single table. Make your table clearly headed. Now, I suggest you draw your table in advance of taking the readings. This way you can fill it up without having wasted time filling up the results and then doing the table. You must include headings. Make sure that your headings have the quantity and the unit. And this unit is consistent with the unit you measure. For example, if you wrote minute here, and you wrote for me 60, that would be clearly wrong. You don't have 60 minutes to do something in an exam. Also, you need to record your raw readings to the same degree of precision and observation, the same level of detail. Now, for practical examination, you will be asked some questions, especially when you are reasoning. you took the raw data, now you need to calculate a few things. You need to show your work in calculations and the key steps to your reasoning. You need to use the correct number of significant figures for calculated quantities. Okay, for graphs, we have an x, y axis. For x-axis, you need to plot the dependent variable. Here. For y-axis, you need to draw the independent variable. Now, when you are choosing the units for your axis, please make it sensible. Please use units that fit for you. For example, take 1, 2, 5 units. Don't 5, 10, anything that you have seen before. 3 units, for example, is not very well done. Now, in order to measure the slope of a straight line, you might be asked to measure the slope or the gradient of a straight line. The gradient should be, so let me write that down. The gradient or slope of a straight line should be equal to delta y over delta x, or change in y over change in x. The last thing that I want to talk to you about is the errors. We have random errors and systematic errors. Whenever we carry out an experiment, there are always going to be errors involved. They might be due to experiment not reading the scale on the measured instrument, which is your error. It can be any type of errors. So, an error might make data too high or too low. So, what do we do to minimize the error? We repeat the tests and take the mean value, which is the average value, to help reduce the effect of random errors. The errors that you need to estimate are not your own. The errors that you need to calculate for are the errors found as inherently in the reading scale. We need to make sure that we have enough good results to present and that our results are as accurate as possible. Now, if you have, for example, a thermometer, and your thermometer measures up to 0.5 Celsius. This means that anything less than 0.5 cannot be measured, and we need to guess what it is. So, this is especially important when we are reading a low temperature, say 3 Celsius. 3 Celsius might be read as 3.5 or 2.5, which is a huge difference for a 3 Celsius. For our 100 Celsius, for example, it won't make a change because it would be 100.5 or 99.5. So it won't make much of a difference. All right, what does that mean for us? How can we calculate the errors in this case? We must be able to calculate the percentage error. The percentage error in instruments is equal to the margin of error, which we can find in our instruments of error, over the actual or mean. Okay, let me write that down another way. So an error percentage is the margin of error over actual or mean measurement times 100%. For example, we talked about the 3 Celsius of before. So this would be our actual or mean measurement. So our temperature, our thermometer measures up to 0.5 Celsius. So the margin of error is 0.5 times 100% over 3, which is equal to 16.6%. Okay, let me just fix that. What about the 100 Celsius? We have 0.5 times 100% over 100 Celsius, which would be equal to 0.5%. See the huge difference? This is all due to this. So remember that when you are measuring for error, when you are measuring for error percentage, there are significant errors that you need to take into account, but you need to take into account your mean measurement or actual measurement and the margin of error. For example, assume you have a graduated cylinder and the graduated cylinder is 100 ml. Your 100 ml graduated cylinder has a plus minus 1 ml. You tried to measure 10 milliliter, 50 milliliter, and 100 milliliter. What would be your error percentage for all of them? Take a moment to think about it. Your answer should be 10%, 2%, and 1%. Okay, try to do it on your own. In the end, I would like to remind you that please, just read the question successfully, try to collect the data and your observations. If you have any tables, draw them ahead of time. Make sure that if you have any anomalous data, like if you have any anomalous, especially when you are making a drawing, if you have any anomalous points like these three, and all of them fit into a straight line, ignore your anomalous points. Remember to make your data layout, especially when drawing a graph, as neat as possible. Also, when you are doing a graph, make sure to put your independent variables on the correct orders. Make sure to label your graph correctly. And when you are finally have finished, make sure to remember what type of apparatus did you use and what was your percentage error in each one. Thank you and good luck in all of your examinations.